Hello, I'm here to discuss with you a recent paper that was published in Bioessays. That work was based on two previous studies, one that was published in Open Biology and the second in Forensic Science International. Man has been fascinated with death since the beginning of humankind, as reflected by art, history, religion, and laws. Not much is known about the biology of death, and that's what compelled us to study it. Turns out that studying the biology of death is also important for forensic science, for solid organ transplantation, and for understanding the response of the body, organs, tissues, and cells to extreme stress. There are many ways to investigate the biology of death. We chose gene expression specifically because genes tell us the physiological demands of the cell. Our study was based on two model organisms, the zebrafish and the mouse. We originally hypothesized that gene expression should stop right away after death. However, we were very surprised to find that this is not true in all the cases. While 99% of the gene transcript abundances decreased with postmortem time, approximately 1% increased with postmortem time. And this occurred both in the zebrafish and the mouse. Shown here is a typical postmortem transcript profile of one gene. Transcript abundances in log scale versus postmortem time. Each data point represents the transcript abundance of an individual mouse. You can see that the points are quite close together for each postmortem time. A maximum was reached in 1 hour, and then transcript abundances decreased. Here is another example of a postmortem transcript profile for another gene. You can see that the maximum here was reached at 12 hours and then slowly decreased. For another gene, you can see that transcript abundance gradually increased with postmortem time. And in this last example, you can see that transcript abundance maximized at 1 and 6 hours and then gradually decreased. What does this mean? 1% of the gene transcripts increased relative to flash frozen controls. Recall that 99% decreased. Second, we can see that there's low variability for the same gene transcript at the same postmortem time, which is indeed interesting. It means that the organisms are responding in the same way in terms of transcript abundances as a function of postmortem time. The third point is different genes have different transcript dynamics, which is surprising. It means that the genes are responding differently as a function of postmortem time. There are two ways to investigate patterns in the data. One is principal component analysis, or PCA, and the other is cluster analysis. Shown here is the ordination plot of the zebrafish, which represents 548 transcript profiles. The two axes of the ordination plot represent 58% of the variability of the data. Each of the data point represents an individual time point. What does this mean? When we assign time points to the data, we can see that the pattern goes around in a circle. Shown here is the ordination plot for the mouse, representing 515 transcript profiles. Overall, the two axes accounted for 98% of the variability of the data. When we assign time values to each of the data points, we can see a pattern also. Cluster analysis also revealed patterns in the data. Shown here are 6 different cluster groups. Each of the cluster groups has a different pattern. The same analysis was performed for the mouse profiles. As you can see, there are also different groups with different timing and different durations. Well, component analysis and cluster analysis showed us that there was a global response in terms of transcript abundances as a function of postmortem time. The next question we were interested in, is there a relationship between gene transcript diversity and postmortem time? Indeed, there is a relationship. We can see that transcript diversity increases in both organisms to 24 hours, then decreases. This suggests that there might be a natural threshold at 24 hours. The next question we were interested in is there a relationship between gene function and postmortem time? Genes involved in stress, immunity, apoptosis, and transport increased in transcript abundance as a function of postmortem time. This is anticipated because this is exactly what happens in response to injury and stress in life. On the other hand, we found many interesting genes that increased in their transcript abundance with postmortem time that we did not anticipate. For example, developmental control genes, cancer-associated genes, and genes associated with epigenetic factors. What does this mean? Currently, we have no adequate explanation for why these gene transcripts increased with postmortem time. It is perhaps just a response to stress. In terms of gene function and postmortem time, some gene transcripts increased as anticipated, while others did not, which warrants further research. Why does gene transcript abundances increase with postmortem time? There are three possible explanations. The most obvious is that gene expression is taking place, and as it takes place, there's an increase in transcript abundances. The second explanation is that some cell types may be more resilient to death than others, so that we may be measuring changes in cell types. The third explanation is that some mRNAs are more resilient to degradation than others. So what we are seeing is a change in resiliency of the mRNAs as a function of postmortem time. This is a scenario of what we think is going on. Shown on the left are live cells in an organ, and on the right, the same organ, but after death. If you notice the blue and the green cells, there is an increase in the transcript abundance after death. The pink cell is no longer around after death, presumably because it is not resilient. The orange cell is still there, but there are fewer transcripts. Earlier, I mentioned that this research is important for forensic science. Specifically, one can use the gene transcript abundances to back calculate how long a corpse or cadaver has been dead. We were able to do this with very high accuracy. Shown here is a relationship between actual postmortem interval and predicted postmortem interval using matrix algebra. You can see that we get a very good fit, close to 1. Shown in gray is the 99% confidence interval. This research could also be important for solid organ transplantation because organs are procured from brain-dead patients. If that patient had been dead for quite a period of time, there could be increased abundance of gene transcripts that are detrimental to the success of the transplantation. A simple way to get around this problem is to design a bioassay that samples gene transcript abundances from organs that are destined to be transplanted into recipients. Before embarking on this research, we had no idea that studying gene transcript abundances in dead animals would be so full of surprises. For example, we found out that gene expression continues for a long time after an organism is dead. We also found out that we can determine how long an organism has been dead simply by looking at the abundances of certain gene transcripts. The last thing we found out was that this research could also be important for solid organ transplantation. As I mentioned, we could develop an assay to assess organ quality based on gene transcript abundances.